Good afternoon, students. I'm Dr. Kusuma Kumari, Professor and HOD, Department of Physiology, MRIMS, Hyderabad, Telangana State. Okay. So today we can explain about skeletal muscle contraction, how it occurs, what are the micromolecular level sequence of events takes place. So before going to actual muscle contraction, we just go through the overview of previous class. What is the structure looks like, skeletal muscle fiber. These are the slides just to remind you and revise so that you can have an understanding of the muscle contraction. So basic structure, the structural and functional unit is called muscle cell or myocyte or myofiber, muscle fiber, which contains many small structures called myofibrils. Each myofibril again contains myofilaments. What are myofilaments? Nothing but again thick filament and thin filament. Thin filament is made up of actin, troponin, and whereas a thick filament is made up of myosin molecules. Okay, actin, troponin, and tropomyosin together is called thin filament or nothing but actin filament. We have got so many other proteins also to keep the structure in a strong and supporting condition during muscle contraction is going on here. Every muscle fiber is supplied by a nerve terminal called a synaptic knob. So usually as it is connecting with the skeletal muscle and it helps in muscle contraction, it is also called motor neuron. Okay, so once motor neuron spreads its action potential by releasing acetylcholine at the junction called neuromuscular junction, it will cause the depolarization of these muscle fibers. So this is one of the muscle cell where sarcolemma with the T tubule and L tubule at right angles they are arranged, and cisterns are the sites for high concentration of storage of calcium ions. Okay. So now we go through what is the structural and functional unit of these muscle fibers is. Again, we go in detail. Myofibril, as I told you in the last slides, thick filament and thin filament. They're arranged in series like this in a single muscle fiber also. Almost cytoplasm is filled with these protein structures. So what is the smallest part of this muscle fiber is called sarcomere. So what is sarcomere? Is structural and functional unit of a muscle fiber. Here, what is a sarcomere is nothing but a junction between two Z lines, and from Z lines there will be actin filaments on either side will be projecting and overlapping the peripheral parts of the myosin filaments, nothing but thick filaments. So here we have read already sarcomere structure, very important structure. So what are thick filaments? Nothing but myosin filaments arranged, almost 200 to 400 myosin molecules arranged like this. Each single molecule looks like a golf club, like a stick with a head projecting upwards in resting condition here, and thin filaments made up of actin, troponin, and tropomyosin. So these two structures, how they are arranged is very important here, which will help in actually shortening of the muscle here. This is overall structure looks like this, where the sarcomere is sliding towards the center on either side. From this side, Z line is moving this side and red line from the other part. So almost the central part called bulky part of the muscle will be shortened by the sliding action of the peripheral parts of the sarcomere here. So this is how, just to show you how biceps is contracting on one side, another one is during extension of elbow, triceps is contracted. So we can see the grossly how the muscle contraction helps in the movement of the joints. So this is briefly, without a nerve connection, muscle cannot contract, to show you how neuromuscular junction transmits the action potential along the sarcolemma through T-tubule. The action potential, when it is spreading, will help in the actually entry of sodium ions into the cell, cause the depolarization. The depolarization wave will be transferred along the horizontally this direction, as well as whenever it is passing through the T-tubule, it will go into the interior. What is T-tubule? Nothing but invagination of sarcolemma here. Sarcolemma is nothing but cell membrane made up of phospholipid layers. Along with the entry of this action potential can come closer on either side to the cisterns. What are cisterns? They are storehouse for high calcium reserve. So when T-tubule along the first step for muscle contraction is a spread of T-tubule action potential when it is passing through, coming closer to the cisterns. We'll try to stimulate some pumps present on the membrane of T-tubule, also on the cistern membrane here. What is cistern? Is nothing but L-tubular on either side, will be enlarged as a small sacs, which will be storehouse for the calcium here. Because calcium is the main ion responsible for muscle contraction here. So we need a release of calcium from here by the action potential spread along the T-tubule. So anyway, this structure of myosin and actin molecule, I have already told you. At resting condition, the myosin head will be just projecting upwards. On activation, it will have two joints, one at the head and neck, and another one is neck and tail part here. So it will be moving upwards to attach to the actin. So around 300 to 500 myosin molecules assemble from myosin filament. One will be having that. So actin also we have to see individually. The actin molecules will have myosin binding site, and myosin will be having the actin binding site. Every seventh actin molecules attached by troponin here. Troponin has got three molecules, I, T, C. And tropomyosin is a ribbon-like structure. Whenever tropomyosin is covering the active sites of myosin, the muscle will be in a relaxing condition. Whenever it is exposing them by moving away the myosin binding sites on actin, then only the initiation of muscle contraction takes place. So for the muscle contraction to take place, we have to move away the tropomyosin. Actually, tropomyosin is attached by troponin molecules. Yeah. So we just go through. Anyway, these are all summary of the previous chapter. So this is about the molecular mechanism of muscle contraction and relaxation. Proper what we have to discuss now. This has got some sequence of events again to take place to cause the smooth contraction of the skeletal muscle. So what are the subheadings we can make? The explanation is excitation, contraction, coupling should take place first, followed by cross bridge formation, followed by power stroke. So all these are very, very important, and here very important role is ATP will play a very important role both in contraction and relaxation, and that is the main energy source for the contraction here. So these are all explained by a special theories which were done before, and out of theories there are many theories, and very at present what is accepted one is called sliding filament theory. Okay, so now we go step by step anyway. So what are different names of theories, and out of which one is reasonable and acceptable is actually sliding filament theory, or also called ratchet theory or walk along theory, which was explained by the scientists Huxley in the year 1969. So muscle contractions work at cellular level with proteins sliding against each other, causing cross bridges, which then leads to muscle contraction. What are the other theories which are not that much came into popularity are continuous filament theory and electrostatic solenoid theory. There are a few more also there. We go through only what is needed at this level is sliding filament theory. So what is the first event is electrical excitation, contraction, coupling. So what do you mean by excitation, contraction, coupling is excitation related with some electrical activity, nothing but action potential along the sarcolemma. When it is spreading, it will pass through the T-tubule, transmission of action potential along T-tubule to interior of the muscle fiber. There we have got voltage gated calcium channels in T-tubule called DHP receptors, dihydropyridine receptors, which are voltage sensors. So stimulation of these voltage gated calcium channels in T-tubule will lead to stimulation. So these will trigger calcium release from ryanodine receptors of sarcoplasmic reticulum, which are present on the cisterns. So which on stimulation of this DHP linked with ryanodine receptors, they will cause the release of calcium into the cytoplasm from sarcoplasmic reticulum. So this is the picture to show you how when action potential, this is a T-tubule passing along this one. This is DHP receptor, which is linked and will be closing the ryanodine when it is at rest. Okay, this is ryanodine receptor, and this is cisterns of L-tubule. So here you can see high concentration of calcium here. Whenever action potential is passing through T-tubule proteins, like DHP gets activated, and this will start opening the ryanodine receptor, which was previously closed and not allowing the calcium to leak out. Once these ryanodine receptors are open, the calcium will start pouring into the cytoplasm here, also called sarcoplasm. So this is called nothing but excitation process, still contraction not started. Because of this excitation process, what is the purpose done is release of calcium into the cytoplasm of this muscle fiber here. Now this calcium will go attached to the troponin molecule here. So when it is coming along this one, DHP, this DHP receptor opens by stimulating ryanodine receptor entry of calcium into the cytoplasm. This is how the calcium released into the cytoplasm will go and attach to the troponin C part of the actin filaments. Once it is attached, this will move away the ribbon-like structure called tropomyosin. This is called troponin-tropomyosin complex, moving away from the actin molecules, exposing myosin binding sites. These dark spots are nothing but myosin binding sites which were previously covered by the tropomyosin. Only when calcium comes only, they just to move away and cause the exposure of myosin binding site. As soon as myosin binding sites are exposed, the activated myosin head on the myosin filament or thick filament will start projecting upwards and attached to the myosin binding site of the actin molecule. Like this, all the activated myosin heads will come and attach to the binding sites of the actin molecules here. So this is the first step for muscle contraction. What is the first step? Calcium coming and attaching to the troponin C is the initial step for contraction. So here this calcium is acting like a junction between excitation reaction and the contraction here. So this is the one joining both the events. That's why it is called as excitation contraction coupling process. And what is the structure responsible for these two events is calcium. So without calcium, this cannot be possible and contraction cannot be possible. So this is the first step of muscle contraction. This is again same picture to show you DHP and ryanodine receptor. If you write at this molecular level, you can score more marks in the exam if you explain the detail of muscle contraction here. And so the same picture shown here, how the action potential shown in the form of star animated and comes and attached to the DHP receptors. And this DHP is when it is transferring and downwards will activate the DHP receptors to cause the opening of ryanodine receptors here. So next process is mechanical event. Contraction is also called as mechanical event. Excitation, nothing but action potential spreading here. So DHP and ryanodine helps in the calcium release, and that event is called excitation contraction coupling. Now we go for the actual contraction mechanism. The first step already we have seen how calcium will come and attach to troponin and unwinding of this violet colored ribbon shaped myosin, exposing the myosin binding sites on the actin here. Once this occurs, the myosin head immediately comes and attaches to that binding site of actin is called cross bridge formation. So this is another important event which will start occurring after the calcium will come and attach. Once this activation will take place by myosin head containing the ATPase enzyme site, where the ATP will come and break down the ATP into ADP and inorganic phosphate, and energy is released. That energy is taken by the myosin head and will move upwards and attach to this one. So for this purpose, we need ATP. So cross bridge formation is nothing but a simple attachment here. Power stroke is nothing but this bending of this head of the myosin at the neck region, pushing the actin filament towards the M line is called as power stroke. For this, also same energy of ATP is needed here. So power stroke to move actin molecule towards the M line. This is the second step of muscle contraction here. Next, formation of cross bridge between actin and myosin, sliding of thin filament on thick filament. Thick filaments are always fixed in its length and position. What will be sliding is only thin filaments around the thick filaments. Usually one thick filament is surrounded by 6 thin filaments here. All 6 directions we have got the myosin heads are present and which can raise their heads, and 6 directions they can attach to all 6 filaments of thin filaments, and they can slide them towards the embryo, leading to shortening called as contraction. So the process of depolarization of muscle fiber initiating muscle contraction is excitation, contraction, coupling. So the depolarization of muscle membrane, electrical response, muscle contraction is called mechanical response. These two responses coupled by calcium ions. The activated myosin head binds to actin forming cross bridge. Inorganic phosphate is released. Bond between myosin and actin becomes strong. This is called as cross bridge formation. And ATP is hydrolyzed to ADP and inorganic phosphate. Energy is released during hydrolysis, reactivates myosin head. Myosin head returns to cocked position. Means again coming back to its relaxation position also. Needing ATP here. So this is one small summary to show you how the cross bridge cycle takes place. Binding of myosin, cross bridge binds to actin molecule. Power stroke, cross bridge bends, pulling thin myofilament inward towards M line. Detachment. Cross bridge detaches at the end of power stroke and returns to original conformation. Binding. Cross bridge binds to more distal actin molecules and the cycle is repeated. That's why it is also called cross bridge cycle. Can be asked in the exam for many times as 5 marks question. So the sequence should be presented in correct order. So this graph will give you an idea of upper one green line represents that this is an action potential which will be nothing but spread of electrical impulse along the sarcolemma is going on, where sodium entry into the cell from ECF to ICF is called depolarization, and a downward line represents repolarization. Means nothing but exit of potassium from ICF to ECF by opening of the voltage gated channel. Together we call it as action potential. So the action potential is very short duration which takes place along the sarcolemma. And after this sarcolemmal event is over, only the mechanical event of muscle contraction, means nothing but sliding action of actin over the myosin, shortening of the muscle takes place. This orange line below graph represents mechanical event here. So the first one what takes place is electrical event, nothing but action potential along the sarcolemma and T-tubule spread. After that only the mechanical means sliding action of actin over the myosin will take place and cause the muscle contraction. This upward deflection is called muscle contraction, downward is called muscle relaxation. Whereas here we call it as upward one is depolarization, downward is called repolarization. So please try to remember both has got different terminology and which event is first means action potential followed by muscle contraction. Nothing but call it as twitch, muscle twitch. So this is again summary of the cross bridge formation. What is sometimes 2 marks, 3 marks question can be asked as cross bridge. So what do you mean by that? You can go through your books and explain that. This is nothing but simple attachment of myosin head, activated myosin head towards the actin. So this is a small summary of the cross bridge cycle followed by myosin power stroke. So this will go on repeat till the muscle contraction we need to continue. Once muscle contraction is over, the muscle has to relax to come back to resting condition. So this is about the power stroke, how the activation of the head swell. During relaxation also coming back to original resting position. What is the first step for muscle relaxation? Is detachment of calcium from troponin and moving the troponin tropomyosin complex over to the exposed myosin binding sites on actin. Once this tropomyosin covers this area, the muscle will go into the first steps of relaxation. So what is the first step? Is nothing but first calcium detaches from the troponin C. Because of this process, the ribbon will go back to the original resting condition and covering all this. Once they are covered, automatically the myosin head has to detach from there and come back to its original resting condition on the thick myosin filaments. So here the movement of head downwards also you need an active process by getting energy from the breakdown of ATP. So ATP hydrolysis is again done by ATPase enzyme present on the head part of the myosin. So relaxation is also an active process. So ATP is again broken down, providing energy to calm the myosin filaments, means recovery stage. So these are the different steps shown here. So this is cross bridge cycle followed by relaxation. So there's a small animation to show you how calcium will come attached to troponin and moving away the tropomyosin and exposing binding sites of myosin, cross bridge formation, and this bending is called power stroke. So overall there is a sliding action of on either side of the sarcomere, where only the actin will be sliding towards the midline, whereas myosin filament length will be constant. So what is the role of ATP is we need three areas. It's main important during power stroke, during relaxation and relaxation, return of calcium. Calcium detached from troponin, where it goes is it should go again to its storehouse located in cisterns of sarcoplasmic reticulum. So it has to go back. For that also you need ATP breakdown. So it is also called active process here. By calcium, magnesium, ATPase enzyme. So contraction is an active process and relaxation is also an active process. So this is how muscle relaxation takes place. Acetylcholine breakdown at the end, pumping back of calcium from sarcoplasm to sarcoplasmic reticulum. Calcium stored in SR with the calsequestrin is the storing binding protein. Detachment of myosin from active is called relaxation. It is also an active process. So already we have seen what are the three sites ATP is utilized here. So once again to summarize, sliding filament theory, ratchet or mock and arm theory is the mainly accepted theory for explaining skeletal muscle contraction. So what is sliding? Actin is sliding over myosin filaments and shortening the sarcomere at the I band and Z line, disappearance of H2M, no change in the length of actin and myosin filaments, especially myosin filaments, accepted theory of muscle contraction. So this is about a summary of the molecular mechanism of muscle contraction, relaxation, nothing but excitation, contraction, coupling, cross bridge formation, power stroke, theories of muscle contraction, role of ATP, in muscle contraction and relaxation, types of contraction, and what is the energy source for muscle contraction is briefly nothing but ATP, muscle relaxation. This is the summary what we have discussed.